the trial, and we are conducting it. So this is the basic difference between traditional and the adaptive design. So in traditional design, you don't have that flexibility, right? When the design is being conducted and you identify, okay, some changes have to be made, some modifications should be made, or you find out that, okay, sample size is not enough, you want to increase it, then you don't have that flexibility to do the modifications. So adaptive designs allow you to look at the data when the trial is ongoing and allows you to make those changes while you're accumulating that data. But we have to remember the validity and integrity of the trial has to be maintained there. So these are basically pre-planned changes. So for example, if you define upfront in the protocol that, okay, sample size re-estimation is done. Okay, so when you conduct the study, when you can do the blinded sample size re-estimation over there, and you might increase the sample size. So what is the benefit here? You will prevent the underpowered trial. If you would not have looked at data, if you would not have done this estimation, you would have got the underpowered trial, right? So this is how adaptive design helps you. It also helps you to stop the whole trial at the early stage for success or lack of efficacy. So, for example, when the trial is ongoing, you might stop it because of some safety concerns, because of lack of efficacy, or some other reasons. So, these are sequential adaptive designs we have. So, this is one of the criteria which you can again pre-plan. Third is abandoning treatment or doses. So when we say abandoning doses, so let's take an example like we have two lower dose, one higher dose, and we have the active control. Okay, you find out during the trial that two low doses are really not efficient. They are not impacting, or we have lack of efficacy there. You might want to withdraw that. So if you withdraw those two low doses, you can continue with the high dose in the reference, right? So that kind of flexibility again you're getting in your trial. It again saves lot of resources for you in terms of cost, time, if you withdraw those lower doses. Then last is identifying the patients who are most likely to benefit. So while the trial is ongoing, we can do some analysis which helps to identify which patients are actually benefiting from this drug, and then we can take that subgroup and then we can randomize them to active and control. So this is the enrichment adaptive design. So I'm just giving a glimpse here. There are multiple types of adaptive design which we can do, and these are really helpful. But again, these designs come with some challenges as well, because there could be some complications with analysis. Designs are really complicated and there could be multiple changes you are doing. So it's little challenge you have with designing part here and with the analysis part. Now coming to like I showed in the agenda, the minimizing bias, right? So what is bias? Bias in the study could be due to any factor which impacts the true value of the treatment effect deviating from its true value, right? So we have a different bias, but I'll be focusing here on the selection bias and the observer bias. Okay, so selection bias is the bias which can occur when while doing the selection of the subject, which subject should get the which treatment group. So for example, like a patient comes at the site, okay, and patient is really critical, so investigator thinks, okay, it's a placebo control study, we will administer test drug to that patient. Now here you see bias is introduced at the selection level itself, right? So to prevent this selection bias, because we don't want investigator to be selecting the patients based on different factors, to prevent this we are randomizing we are doing the randomization of the patients where each patient will have the same chance. Okay, so for example, you have test and control and you have 100 patients, each patient will have equal chance of getting test and control. There is no inclination towards one group or the other. Another importance of randomization is it ensures comparability between treatment group, because most of the baseline characteristic before randomization they are similar. That's why when you do randomization, it helps or ensures the comparability between the treatment groups. Then there are different types of randomization we can do, and again, it will depend on the requirement and the study design. So here we have simple randomization, block randomization, stratified, and adaptive randomization. So talking about simple randomization, so for example, if there are two groups, okay, you have A and B. Now this subject A or B, they have constant probability here of being allocated to either of the group. Okay, so for example, if you flip a coin or use the random number tables, so here you can see here we have 10 subjects. So each subject will have constant probability here that they are given either the treatment or the control. Now, what happens here? What is the disadvantage you can see here is when the sample size is really small, you might have the imbalance. But if you increase the sample size, let's say it's 500, 400, then if you use simple randomization, there will be approximately equal balance. But in smaller sample size, this is a disadvantage of the simple randomization. Another is the block randomization. So, for example, your sites are competing, they are doing the competing enrollment, and you want there should be a balance between treatment group at all the times during the study. Okay, so for example, after 1 month of enrollment, you don't want that there are too many subjects in test and too less subjects in placebo reference. So you want there should be some balance between the treatment group. How you can achieve that? You can do that by the block randomization. Okay, so what happens in block randomization is that you create the blocks, okay, and within the block, subjects are randomly allocated to the treatment group. So if you look at the picture here, we have like 6 blocks here, right? So within each block, we have four patients, and 2 patients are randomized to test, and 2 patients are randomized to control. Okay, so this is how we do. Now, if you wonder what should be the block size, so this block size, first of all, should be decided by a statistician in the trial, and it is again based on your sample size and the number of treatments you have. Because, for example, if you have a sample size of 100 and you have, say, 2 treatments, so you can take the block size of 4 as taken here. If you increase the block size too long, let's say you take 8 patients block size, then you might have again imbalance. So the whole idea of doing block randomization is to avoid that imbalance, right? So you don't want to increase the size of the block. And what happens if you make the block size very small? Then you can predict the next allocation. So the block size has to be decided very efficiently depending on the sample size and depending on the number of treatments you have in the study. Now coming to stratified randomization. So stratified itself, the terminology says that we are stratifying it, right? So it balances the subject based on the baseline covariates. So baseline covariates. So covariates could be here gender, age, race, disease severity. And again, we cannot choose it randomly. It has to come from the clinical judgment that, okay, based on this covariate, this can impact your actually the variable you're studying, the dependent variable or the primary objective you're studying. It can affect the treatment response. So based on the clinical judgment, you pick some covariates, one or two, I mean, based on important covariates, and then you stratify the subjects based on that. You create the strata. So for example, if you have gender, let's take example here, we have gender as a covariate, so we stratify based on male and female, and within each strata we are doing the randomization now. Okay, so if we do over stratification, so for example, you stratify by gender, you stratify by age, race, everything, then you will have over stratification, right? And in that case, what could be the disadvantage? That you need to have sufficient sample size, that you have enough patients within each stratum. So here we have to again look into the important covariates only. And another disadvantage is it can be only done for the known confounders, right? If you don't know any baseline variable or the confounding variable that could impact the response, you cannot take that into account. Okay, so this is how you do the stratified randomization. Then moving to a very interesting another type of randomization is covariate adaptive randomization. Now, till now, we have seen that we generate the randomization schedule, and we do based on block randomization or based on some strata. But here, the subject is allocated or assigned to a particular treatment based on the specific covariate and the previous assignment. Okay, so at some point of time, if we already have some subjects in the study in test and control, so when the new subject comes based on the covariates and based on the previous assignment we had, we do this randomization. So if you look at the first table here, and I'm going to discuss here the minimization method or the frame method here. So if you look at the first table, we have two covariates we can see. One is the blood pressure, where we have two categories of pre-hypertensive and hypertensive. Other is the age group where we have less than 65 and greater than equal to 65. Okay, so at some point of time, for example, if in drug A is given to 12 patients, which are distributed across these covariates, and 8 patients are given drug B, again distributed across these covariates, what we will do is when one patient arrive, we will assign drug A to that patient. Okay, so what happens here is if suppose that patient is hypertensive and less than 65, we assign it to drug A, and this count you can see 8 has gone to 9 and 5 has gone to 6, whereas in drug B it's 3 and 2. Okay, so we calculate here the chi-square goodness of fit test statistic. So the chi-square we get for blood pressure is 3, for age we get 2. Okay, so the maximum chi-square we pick from here, that is 3. Again, we assign that patient to group B. Okay, it's not like we just randomly assign to drug A or B. We assign to both the drugs, we calculate this chi-square statistic, and we pick the maximum chi-square statistic for each drug we have got. Whatever is the minimum, then we assign the subject to that particular drug. Okay, so here you can see based on the covariates, based on the earlier randomization of the subjects, we are doing this assignment to the subject. So this is the frame method. So terminology itself says that based on covariate, you are adapting it and then you're doing the randomization. Okay, so next is minimizing the bias, another kind of bias, and that is through blinding or masking. So what could be the other bias? Let's go back to the same example we were discussing during randomization. Okay, now the subjects have come to site, and investigator know the subject is given placebo and another subject is given test. Okay, so investigator since knows that what treatment has been given to which subject, investigator might be biased in taking some actions towards the subject. Okay, so maybe they might pay more attention to the subjects who are given test and do not pay more attention to subject who is given placebo, right? So that kind of biasness are being introduced in the study when investigators are aware what kind of treatment is being administered to the subject. Similarly, I'll show in subsequent slide that how we get the benefit if blinding takes place. So here we have two type of blinding. In single blinded study, if you look at this picture, only the subject participant is blinded. Okay, but the researcher is aware about the treatment group. Whereas in double blinded study, both are blinded. So neither they are not blinded. So they are not aware of what treatment has been administered to a particular subject. Now, let's look at what are the benefits. So, generally, we say double blinded studies are gold standard, right? So, why do we say that? What is the benefit if participant is blinded, or the trial investigator is blinded, or the assessor is blinded? So, when participants we say are blinded, then it's less likely to have the psychological impact. So, for example, if I'm aware I'm being given test drug, and it's a new drug, so new always means improved for us. So we'll be like, okay, this drug is working, and we might have some psychological impact on the responses we give. So for example, if the investigator asks me, are you feeling better, even if I'm not, I will have that psychological impact, and I'll think, okay, I'm given the better drugs, so maybe I'm feeling less pain now, right? So that is how psychologically it's impacting my responses. Then participants are more likely to comply with trial regimens when they are given when they are blinded, because they're not aware which drug they are given. So they are less likely to leave the trial, they are more likely to comply with the trial. This is like this is the just glimpse that if participants are blinded, then how it is beneficial. Okay, another is trial investigator. Now, as I mentioned earlier, trial investigator, if they're aware of which treatment is administered to the subject, they might transfer their inclination, they might transfer their attitude to the participants. They might differently administer, or differently take decisions regarding the participants. They might differently withdraw the participants, adjust the dose, or encourage or discourage the participants to continue the trial. Okay, assessors, when I say assessors, it could be sponsor staff, it could be the vendors who are involved in clinical evaluation. So if they are also blinded, it's less likely to have the bias regarding the outcome assessment. So this is how we can see if participants, investigator, assessors all are blinded, we are minimizing the observer bias or the measurement bias in the study, and that's why it's called more of a gold standard. Then coming to the changing perspective of clinical trial designs, we have already discussed that adaptive designs, how it's helpful. Then I've already covered use of historical control part. But yeah, the RCTs, these trials are really time consuming, these are expensive, and we really want that with innovative approaches, we make this process more faster, efficient, so that drugs or the medicines can reach the patients to as per the requirements. So here we are doing different kind of things now. So adaptive I discussed. Now coming to real world evidence. Okay, what is real world evidence first of all? So now with the advanced technology, okay, with the we have this wearable watches, we have biosensors, we have mobile devices, computers. So there's lot of data, right? If you look around, you go to a hospital, you see a lot of data is getting captured in computers. So this real world data we have in abundant now. right? So if we use this real world data and the clinical evidence we generate using that, that's called real world evidence. Okay. Nowadays real world evidence is really helpful to establish the hypothesis for research. It's also being now getting involved in the drug approvals also, so regulatories are also considering such kind of studies for expedited drug approval process. And one very good thing about real world evidence is that you are considering the actual practice, right? Because when you are doing the control trial, it's like you have some inclusion exclusion criteria, you have more homogeneous population. But when you are doing real world evidence, you have the heterogeneous population, and if whatever you're testing there, it's in the actual pragmatic approach. So that is how we are moving towards real world evidence. And there is a lot about this, so I'm just touch basing that how we are moving towards it. And another is precision, or you can say personalized medicine. So in medicine, what does it follow? It says one size fits all, right? That means one treatment that can be used for all subjects for that particular disease. But is it really possible that when we apply this practice in the real world or the heterogeneous population, it might actually work, especially for the diseases which are very rare? So research has shown that humans really vary in terms of genetic, their physiological, biochemical behaviors, and therefore it's important that we tailor our medicines now, right? When I say we tailor our medicines, that means based on patient to patient requirements, we have the medicines which meet their medical needs. Another is now master protocol. Nowadays we are really hearing this term, and master protocols are being developed. So but what is it? And as we say, it's master, right? So it is answering multiple questions. Okay. So in master protocol, again we bifurcated based on umbrella, basket, and platform. So in umbrella protocol, we are studying different treatments. So we have same particular disease, and in that we are studying different treatment. Then it's called umbrella protocol. When we have one treatment, but it is studied in different related diseases, then it's called a basket protocol. Okay. And the third one is platform, where we are studying different treatment in the same disease, but then treatments has they can enter or exit based on some decision algorithm. So these are different kind of protocols we have, and it is really helping to answer multiple questions. But it again has its advantage and disadvantage. If we look at the cost perspective, if it is done the partnership kind of method in the cost partnership, then it could be cost effective. But if it is done by independent organization, it could be really costly for them. Another advantage of master protocol is that it brings together a lot of stakeholders, because you are studying sometimes multiple diseases, you are studying multiple treatments, so it could be from different companies, so it increases engagement between the different stakeholders as well. Okay. So master protocols again have its advantage, disadvantage, but it's really really helpful, because we are able to use the same infrastructure, same kind of study, and logistic issues are also very less in master protocol. But then it has pros and its cons. So this is how the clinical trial designs, the what we have been doing, like from RCTs to real world evidence to adaptive, now the tailored medicines. So I'm just trying to highlight here how the landscape is changing within the clinical research. Okay. So just now here to conclude, the critical component for this for any successful design is the collaboration. Now for any work, for any company, any kind of area, obviously collaboration is important. But I want to emphasize here that at the designing phase, if there is not a proper collaboration between different stakeholders, we might miss out on something. And every stakeholder, whether it's from statistics, from data management, or the medical team, or regulatory, everyone has their own expertise, right? So everyone brings in their own expertise and shares the ideas when designing the study. And therefore the collaboration is really really important at this stage of the trial. Another thing I just want to highlight, I think it's already highlighted in earlier session also when we had from Mahendra, that we should involve statisticians in very early stages of design. Right, we should not go at the end of the trial and we ask them, okay, now we use different statistical methods and we try to overcome the design flaws. No, we should involve statistician from the very start. We should not wait at the time of data analysis. So statisticians are just not there to analyze the data, but they can help you at the study design. And it could be either, as I told, we want to minimize the study, we want to do randomization. For randomization, if you're using block randomization, from where the block size is coming, it's coming from statistician. If you want to do blinding, then it's really important that very clearly you define in protocol that who are blinded, in which scenarios the blind would be broken, and if it is broken, then what is the standard operating procedures around that, it's been established or not. So all those details have to go very early at the design phase. And statisticians can help you with the sample size. Definitely, I'm not going to speak much about it, but yeah, sample size, if you make it very large, it is again a waste of time and resources for you. If you make it very small, it might lead to the inconclusive results, right? So sample size also has to be very adequate for the study design, and statisticians can help you with that. So, just to conclude, I would say that it is very difficult to fix a poorly designed study once you have implemented it, right? So, once study design is implemented, there's nothing you can do about it. So, we have to be very careful from the designing stage. And once the designing stage is completed, the challenges lies mainly in the data collection, correct analysis, and the accurate reporting. Okay, that is all from you. Thank you so much. Any questions?
Well done. Thank you so much, Neha. I think we are pretty much on time. Okay. So, thank you so much for listening. And I could see several questions in the chat and in the comment. So I tried to collate those things, and some of the questions I answered on the chat. And I'm not very good in typing so fastly. So I put things on or your question on a Word document. If you allow me, I can share the screen, and then you can take, or we can take those questions as well. All right. So hope you can see my screen now. Can someone confirm if you can see my
Yes,
screen?
yes, Mahindra. It's
Oh, you
visible.
can see? Okay. So now some of the questions on the chat. Let me hide it. So those questions were answered. So what is M1? So M1 is basically the pooled effect of or 95% CA from the historical data, and that we need to get it close to the null effect what we would like to estimate, and put a reference here if you would like to read more about non-inferiority, how it is calculated and so on. So it's there. Then say, okay, so non-inferiority, again the question is, can you please explain this again? Non-inferiority margins are calculated in two ways. So either you can first do the meta-analysis based on the historical data, get the effect, and from that effect, that is your M1, and then what you would like to retain as your effect size when you run the trial, so that becomes your M2. So fraction of M1 you take as the non-inferiority margin in M2, and that's how you use it. Sometime you can use the lower limit of 95% confidence interval from your meta analysis, and that also you can discuss with clinician and get the effect size that they would like to retain in the new study. Okay, how sample size and randomization is varying compared to BAB and clinical trials, because CT using ratio 1:1 and didn't get clear understanding for calculating sample size in clinical trials. So sample size part we will have the next session after the break. So we will try to cover there regard and say, okay, your car is not better than that one, or this car is not giving mileage better than that car. So it gives, or it helps us to take a decision which one is better, and this is where you need the comparison. Without comparison it doesn't help. Yeah. uh, how to select parallel or crossover design, and what are the parameters to be considered? This depends on which phase of drug development you are. If you are running a phase 1 study on PK PD endpoint, maybe you could consider crossover study, because you don't want to expose more subject on the trial or the experimental drug. And in crossover, because same subject play a control of itself, so you reduce the variability, and this is why maybe you prefer crossover. But when you run a phase 3 clinical efficacy and safety study, then you have to have a control, and in that case the regulatory requirement is that you have to have a parallel group study. You can choose different randomized ratio on other part of the trial, but yes. Adaptive design is accepted by all regulatory agencies. It depends what type of study design you are proposing and what indication you are looking in. For example, during the COVID, there were so many vaccines studied, and there were different type of designs used: phase one, phase two, seamless, phase three, and so on. So regulatory agency do not have any choice or a particular design, as long as we are able to answer the clinical questions. Non-inferiority trials generally sample size is low, around 100 to 200. No, it depends on what the endpoint and what the non-inferiority margin you are selecting. It could be 2000, it could be 3000. It depends on the clinical question and also the clinical difference that you would like to detect. And superiority trial is quite larger. Yes, in general, sample size conducted in non-inferiority and equivalence are higher compared to the superiority trial. Remember, in the mathematical calculation, the clinical difference comes in the denominator, and when you have superiority, means your difference should be greater than zero, then you have the absolute number. But when you go to non-inferiority stuff, in that case, you need to consider the non-inferiority margin as well. So you are going beyond or below zero. But that we will show when we do the sample size part. Then washout period in practice or in general, we consider based on the half-life of the drug, and that will, if you consider say 5 times of half-life, drug will get eliminated from the body, and that's you pick as a half-life or your washout of the drug. Why we use placebo? First thing, you need to show that your drug is better than placebo, because placebo itself has its own effect, and in some of the indication, placebo effect is more than 20%. So if the 20% difference you would like to detect in your trial, and the 20% difference is observed in the placebo, then actual treatment effect is zero. So you need to show that your treatment effect, once you subtract from placebo, that's the actual treatment effect that you would like to study. So we need placebo. In non-inferiority slide, placebo as a reference. So now would you like to answer this?
Placebo is reference drug. No, so we had test, reference, and placebo all three. So when I was showing the non-inferiority example, we were establishing that test is non-inferior to reference drug, and we also established that test and reference are superior to placebo. So we
Yeah.
are not establishing the non-inferiority to placebo here.
Yeah. So you need to prove that your drug is again better than placebo, superior to placebo, and then it's non-inferior to the
Yeah.
study drug. Okay, and then there is a answer also: to showcase superiority. First, we need to prove non-inferiority. No drug versus test in case of absence of active comparator. One can pick up placebo to provide base for comparison. So active is needed. Then how analysis is complicated in crossover design versus the parallel design in RCT. Now I think analysis remains complicated even in the parallel design. Also, some of the stats analysis are very complicated. As long as you know the answer, or as long as you know how to do the data analysis, then all the analysis are simple, not
Yeah, I think just
complicated.
to add here, when
Yes.
you construct the tables, right, the outputs and all. So in parallel design, it's quite simple. You just construct the table based on test and control. But in when you have the crossover, sometimes again you have a discussion medical team, and some tables we want to be present based on sequence, some you want to present based on treatment. So from that perspective, it's a little complicated at initial phases that how you want to represent your outputs. So not as such analysis. Analysis is difficult here, but yeah, if you know how to analyze the data and how to present that data, then it's okay.
Perfect. So there's next question: what analysis has to be done in crossover study? Is it paired sample test? One thing very important: how you calculate the sample size. That helps you to decide what analysis method you use. But most of the time in crossover study, the analysis method is used based on the analysis of variance, where you consider period and sequence, and mainly your data is related to PK. So you have log transform data, and then once you complete the analysis, then you back transform to the original scale. uh, then blinding of drug to subject is advantage or disadvantage? Yeah. Blinding, why we do blinding? We would like to reduce the bias, and if subject know that what drug they are taking, maybe there is a psychological effect, and that could also have impact on the outcome. So I think it's good to have double blinded. uh, um, then this basket trial umbrella, it's very big topic, but I think it's good. We can use some references, and if you can add, so people can read when they get some time.
Yeah.
Okay, is there any tool available for minimization randomization? Minimization of randomization. So minimization is a process of randomization, one of the process of randomization, and there are techniques, tools. There are a lot of literature available on how to do the minimization randomization scheme, and there were a lot of things considered that why we do all the time this standard practice of permitted randomized or block randomization. We can use minimization technique as well, but it depends, and also there are advantage and disadvantage of one or another techniques or other. So scientifically we need to decide which one is suitable for a particular design, but yes, minimization can also be used, and there are a lot of ways of doing randomization or the minimization randomization as well. Okay, so these are the question on chat, and I think now people are still adding questions here. uh, okay, deciding whether okay, what is the basis of deciding whether we want to demonstrate non-inferiority or superiority? Very good question. So how you design the study, if you feel or if your clinical have the feeling that your drug is better than the existing drug, you can decide the superiority. But if you have 0.00% doubt, you say, okay, I'll prove the non-inferiority, and once I establish the non-inferiority, I can go and test for the superiority as well. So this is allowed in clinical trial, but vice versa is not allowed. So if you set a study for superiority testing, and if you fail, then you can't test for non-inferiority. Okay, so this is how we need to see the things. How block size is determined? Is it based on the sample size? Yeah, block size usually is 2n, so the number of arms you use multiply by 2. If you're two treatment in a block of maybe you can use of size 4, so one treatment is repeated twice. Again, depends on how large your study is, and then how many centers you are using in the study, so that also have some insight, and that needs to be considered while defining the
Also
block
the
size.
ratio,
Also
also
the
the
ratio.
randomization ratio, yes.
Yeah. Mm, please explain when to use placebo and in which phases of clinical trial. So placebo, if there is no critical condition of the study participant, I think it's allowed to use placebo. But there is an published FDA guideline, the selection of controls. So in that, a lot of details are provided. Most of the oncology study, maybe you don't want to use placebo, but if you have a studies like migraine or an early phase like phase 2 study or something like that, you can use placebo just to see the treatment effect, and it's allowed. But again, we need to be very careful on selection of the control, and then it needs approval, and also discuss with health authority if they allow to use placebo. Yeah. How block size we discussed. Then how block randomization different from simple randomization. Per my understanding, it is still 50% chance of allocating a test drug or a comparator. Okay, so now,
Yeah,
Rupina, would you?
here, yes, I can give an example here. Okay, so if you have just a simple randomization generated, okay, so when the study is ongoing, it's possible that first 10 subjects or 15 subjects, there you have more uh, subjects given to test arm as compared to the placebo or the reference drug. Okay, so at any point of the trial, you will not have that balance between the treatment group. Okay, whereas when you do block randomization, it's not just you are creating the blocks and you're randomizing within it, but when it goes to site also, then investigator is randomizing the subject, then they have to first exhaust one block. Okay, so before moving to the another block, they have to exhaust the first block. And within the block, since you have randomized as per the ratio and the balance is maintained, then while the trial is ongoing, you have the balance between the treatment groups. So that's the idea of using block randomization over the simple randomization.
Yeah, and just to add, simple randomization. What do you mean by simple randomization? If you toss a coin and then take a decision, if it's head or tail, then if head, then treatment A. If tail, then treatment B. If your sample size is 200, do you believe that if you toss coin 200 times, will you get 50% head and tail? No, of course not. You can try, not for 200 times, but just try for 10 times, and try the 10 times 4 times, then it will be 40 schemes you create, and see whether every time you are getting 50% chance of being allocated to one of the drug what you are looking for. Perfect. Now the next question is, what is the basis of deciding whether we want to demonstrate non-inferiority and superiority? I think we answered this. Placebo is a dummy drug. Then in clinical trial field, can we check mental or emotional health? Okay, this is very difficult question. Very difficult to check mental and emotional health. And one of the thing is in disease area like Alzheimer's and Parkinson's, how you test the dummy drug effect. Okay, and sometime psychological effect works. If you go and ask a patient, okay, you are feeling good, everything looks fine, and you will get well soon, and in these many days you will be discharged from the hospital. They have that the mental implication on their health as well. If you though I'm not a psychologist, but if you consider that, okay, if you feel I'm tired and you feel like you are tired. Okay, so the mental itself has its own effect on your human or on the body. So it's very important, but I think it's really critical to see placebo as a dummy drug, and it is given in neurological disorders. The placebo is used as a control because it has a very high effect. Okay, elimination rate, or KEL. KEL is the concentration elimination is better to calculate in funny. These are different
Phoenix.
softwares. See, software is not a machine. Software is have inherent statistical or mathematical calculation. If you know how to calculate manual, you can do the manual calculation. Because if you say how to calculate area under the curve, or AUC, when we calculate it, it's a trapezoidal rule, is a mathematical concept. So the same thing is inbuilt in the system. You don't have to do calculation manually, but you can use Phoenix as well, and some other softwares. In SAS also you can do, in R you can do. So there are many ways of calculating this. It's okay. How to select non-inferiority margin, I think we discussed. uh, okay, we know that study design is formulated based on many parameters, but which kind of design are more accurate and more preferable for clinical trials? Okay. It is basically a decision between statistician, clinician, and the operation. Okay, when you design a study, how feasible the study is to conduct, how, or will you able to answer the clinical question that you are trying to answer, and some of the other aspect, money matters. So every clinical trial has a lot of investment, and that investment also have influence on that what type of study you consider. But science takes priority over all other factors. Okay, so yeah, so sometimes you have, say, endpoint based on the proportion, sometimes you have endpoint based on the survival based endpoint, sometimes you have endpoint based in change from baseline, and so on. So it depends on what the study or indication you are in, and what is the question we are trying to answer. So based on that, we decide that which type of clinical trial is preferred over other. And we usually have the justification in the study protocol. If you read in protocol, there is a mention about selection of design, selection of controls, randomization, blinding, everything. All right, what if results of test drug is superior during non-inferiority trial? In this case, will authority accept superiority? If you state in the protocol that once I establish the non-inferiority, I will test for superiority, authority will accept it. Yeah, if he's written, is done. If he's not written, it's not done. Are there any new technique implemented in randomization and stratification using AI? Yeah, at least I'm not aware. And I think the concept will remain the same. Again, AI is a concept, and then you have machine learning. Those are tools. So if you give some instruction to AI will fetch some information from somewhere, and then they will create the randomization and stratification. But the concept will not change. When do we use relative risk difference to calculate M2? So when your endpoint is based on relative risk, then you use relative risk based endpoint. If your endpoint is based in change from baseline, then that will be your endpoint. If your endpoint is based on odds ratio or the proportions, so based on the endpoint, the calculation will also vary. How do we calculate non-inferiority margin for a vaccine trial? You don't have to worry. because the regulatory agency is nice enough. They provided us the non-inferiority margin of 10%. So if you read the guideline where it says non-inferiority margin for vaccines trial, so it's a 10%. Similarly, if you are running a study in diabetic care, so the non-inferiority margin for SB1C percentage change is 0.3%. So that is predefined by agency. So we don't have to justify that. We can use the same. If you would like to define something different than what is specified by the agency, then you need to provide the justification that how you derived the non-inferiority margin. All right. If next question is, if population is large and vaccine is not known, when which statistical test we should take to determine if significant difference between means of two group and variance is not known. Okay. How we design the study. If variance is not known, then maybe sometime you use the similar indication or the historical data, and you try to estimate the variance. And if you are still not sure, then what you do is, maybe once your study is started, you say after 50% or 100% or participant enrolled in the study, you calculate the change from baseline at certain visit, and then you try to calculate the variance, whether you are too far from your assumption. So, and still there are the blended sample size re-estimation techniques are there. You can still use those techniques, but that needs to be defined in the protocol. Okay. Now some questions I took from the chat. If you can see my screen, why do we need comparator? I think we answered. Washout, not 95% say always. It can be any of other level significance. Yeah, you can use 50%. Then you don't need to run a trial. You just toss a coin and take a decision. But the 95% CI is something regulatory agency is looking for. Now, if you ask, okay, so in bioequivalence study we use 90%. But the 90% are like two one-sided tests that we use. So one side 95%, another 5%, other side is 5%. So 95% is something coming from the agency. And if you are looking non-inferiority or superiority kind of stuff, still you can change it. Maybe you can have 2.5% one-sided or so. Uh, how analysis is complicated, we discussed. Non-inferiority margin, we discussed. Point estimate, this is we discussed. Yeah. So need a comparator in clinical trial. As I mentioned, ICS guideline you refer, and it talks about the selection of the control, and that will provide you more detail. This is very interesting question. Let's imagine that clinical trial is ongoing on a new drug for indication XYZ, for which we already have a gold standard in market, but one of the objective is this drug is more efficient when compared to the golden drug. So when we analyze, if the result is equivalence, then what will happen? This drug will be approved. No, so your hypothesis is what you want to say. Would you like to prove equivalent, or would you like to prove better than that gold standard? So based on that, you need to define your null hypothesis and alternate hypothesis. If your alternate hypothesis is accepted, you can claim for that. If is a equivalence, we have that things, different drugs available in the market, where uh, where the biosimilars or bioequivalence, or those kind of studies are conducted. So those are considered to be a similar treatment to the existing ones with a certain margin. So again, you need to have a biosimilar margin, so plus minus 2%. If that is falling within, the confidence interval is falling within that criteria, then you can claim that my drug is as similar as the existing one, and you can market. In oncology disease area, in arthritis, in psoriasis, there are many treatments available. Those are like biosimilars, and because the cost will be low, so definitely regulatory agency will look into those aspects as well. Now, different type of randomization, so I think they have covered on those. One question is about why we use 80% to 125% for bioequivalence limit, and where these numbers comes from. So FDA basically consider that plus minus 20% difference in the systemic drug exposure is okay, and that is not clinically significant. We all know that PK data is not normally distributed, and we try to take the log transform the data, and then we do the analysis. So when you consider log transform, then how that number falls. So the 25%, if you have 25% on log transform scale, where you see the the symmetrical distribution around 100. So this is how this data looks like. This is where 100, it means there is no difference, is 0 on log scale, because your ratio is 1. Now you take log of zero point 0.8, that becomes minus 0.223. If you take log of 1.25, this becomes 0.223. So this is where your distribution is symmetrical, and that's why these ranges of 0.8 to 1.25 are used in the bioequivalence type of setup. I think with that, we tried to answer most of the question, and thank you so much. All right, so now we are close to the break time, or we are over?
Preeti, you
Previous
are on
slide.
mute.
Now, am I audible to you guys?
Yes, yes, yes.
Okay, good. So we are almost, we have completed break time, but let's push it a little bit. Yeah. So
Okay.
instead of 15 minutes, let's take 10-minute break. And I'm putting a slide for the upcoming session. So if you come back early from break, please see what is upcoming session. That will be useful.
Yeah. So we'll
So see
start
you guys
at
at 10:10.
10:10. Okay.
See you
11:10.
guys. 11:10.
11:10.
Yes. Thank you,
Yes.
Mahendra.
Yeah.
Thank you.
And keep your lines open. Please don't disconnect. Huh? Let's have some morning flute by the time people will join. Very good. So Arti, you are starting first or Suban starting first?
I'm starting first.
Oh, you are starting first. Very good.
So do I start sharing the screen, at least the main window?
Yeah, just hold on. Just hold
Okay.
on.
Recording in progress.
Okay.
Participants are also in. Yes, yes, they will. They will.
Very good. Very quick solution. I think we could see participants coming in. Just allow a few minutes, please.
Okay.
Three patients per group or in total?
Yes.
Okay, let's try something different. What type of measure are you using? And is this an in vitro study? An in vivo mouse study?
It is an in vitro study of patient samples, and we are looking at measures of apoptosis after treatment with drug A and drug B versus drug A and control. And drug B alone.
Oh, how many groups are you analyzing altogether then?
My grant is due tomorrow. I just need to know if I can just use 3 patients per group.
Okay, well, you probably should have come to me earlier for help with this.
All I need you to do is tell me that 3 patients are okay.
So, it is not that easy. Your marker for apoptosis may have a large amount of variability associated with it that could make it difficult to ascertain differences based only on 3 patients per group. It also depends on what kind of outcomes you are looking at. Are you looking at normalized changes in expression levels from baseline? 3 patients may not be sufficient to detect differences in these expression levels between the different treatment groups.
I do not understand.
I'm saying that you may need more than 3 patients per group, but that I need more information to determine that.
The serine protease granzyme B is made in cytotoxic lymphocytes, where it is inactive due to the low pH of the storage granules. It is measured immediately upon delivery into target cell cytoplasms, where the pH is neutral. This is followed by cleavage of procaspase 3 by granzyme B in the target cell cytoplasm and the subsequent induction of apoptosis. All proteolytic events can be quantitated at the live single-cell level by flow cytometry and imaged by confocal microscopy.
I understand very little of what you just said. Maybe we should talk more about this.
I just need to know if 3 patients are enough. My grant is due tomorrow.
We will need to meet to better understand this before we can determine the appropriate sample size.
I can put you on my grant for 0.5% FTE. You can design and analyze the data later.
Do you really think that is enough to do the work?
Well, it may be less if it is awarded and the budget is cut.
Please go away.
So now you must have heard why the sample size determination is so important, and the discussion between a statistician and a researcher, how critical it is to understand each other's language. And with that, I'll ask Aarti to share her screen and take us through this sample size estimation for different scenarios. Over to you, Aarti.
Thank you, Mahendra.
Yeah, you can put your slide on slideshow mode.
Is it visible?
Yes.
Okay. So welcome everyone. So I am Aarti, and today I will be taking you through the sample size estimation, basic principles, how it is being calculated, what is the importance, and I will be taking you through towards the end while calculation of the sample size using certain softwares. So before I start, this is the disclaimer which mentions that the views expressed in the sessions are those for the author and not necessarily reflect the views or positions of any entities that I am presenting today. As I mentioned that my agenda today would be just to explain you and make you understand that what is a sample size, how it has been calculated, why is it so important for calculating sample size in any clinical trial, what are the important components of sample size estimations, and I will be taking you through certain calculations for certain clinical designs. So with here I start like what exactly is a sample size in research. So all we know that when we conduct a clinical trial, we are basically trying to answer any of the questions that we have in our mind. And so the number of observations used for determining the estimations of a given population. So here the picture is presenting in front of you is showing that there is a target population from where you have to withdraw certain number of participants in your sample. And why is it this important that we have to do this? Because for any clinical trial, when you have the certain eligibility criteria with patients with different disease conditions or with different style of aging, or it is gender wise kind of a study, so you need to first of all target which kind of patients you require in your study. But you cannot study the entire set of population that is existing on this planet. So what we do is that we withdraw some number of patients of that particular characteristics, and then we have the sample ready for our clinical trial. Now, why do you think that there is a need for this kind of a sample size estimation? So it is nothing but there is a necessity that you cannot, as I mentioned, you cannot study the entire chunk of population with that particular disease condition. You need to have certain number of patients in your study in order to achieve your goal. Also, it is not practical to have each and every person with that particular condition to be enrolled in the study, invest in your money. So finally, doing a clinical trial is also a business where you could not have each and every person being involved with that particular disease condition or with that particular characteristic. So it is not practical to do so. Hence, you need to do a sample size estimation. On the other hand, it is very cost effective in a way that if you have certain number of patients, you will involve the money into it in that respective patient population only. And of course, manageable. So in a clinical trial, there are different stakeholders, including who are investigators, coordinators, statisticians, and many more who are part of this particular clinical trial. So that is something which is it. But beyond this of things that are presented on your thing, why really still we need to do the sample size estimation? Can't we still go ahead and decide on a number? Now, if you look at statistical, when we have our theoretical studies, when we did on statistics, it mentioned that sample size of 30 is still considered to be a very large sample. So why not use that? But there is a basis to this is the guidelines that we need to follow, which is the ICH guideline E9, which talks about the sample size estimation, what should be the number to be presented, has to be decided right at the beginning of the clinical trial being in plan. Another is the regulatory requirement. So today I'm just presenting something which is on Schedule Y, which is presented under Appendix X and Section 14, which presents that you need to calculate the sample size and you need to determine including all the assumption estimations, and you write the statistical report in your protocol. Now, when exactly we want to do the sample size estimation in clinical research? So calculating sample size is always before any of the intervention is running, in the way that there is not a single treatment dose has to be given to the patient unless you have decided what should be the sample size. So you need to see that whenever you start your clinical trial, before that you have the plan in place. So your protocol is basically the blueprint of your study, and in that only, when you have investigators, you have statisticians taking the insights from both the things. So you know that for any clinical trial to be conducted well, both statistical and clinical significance have to play an important role. So a well designed study will lead to a valid conclusion. That is what is the final I would say the key point in this particular sample size estimation. Now, why we do not want to fix it to a specific number? Why really we want to go and estimate and calculate sample size, involve statisticians, do so much of research, do literature search for getting the estimates and then do the sample size? So just giving you an example, suppose you consider a sample size which is very small. And after conduction of the trial, you get to know that you are doing a hypothesis testing, and at the end, the p-value is coming out to be non-significant. This could be the reason. One of the reason would be the sample size that you have chosen a very small sample size. So it will produce inconclusive results, will have no valid learnings, means you might not get something that you had started for. So your answer to the research question is not answerable at that moment. So it is going as a waste of effort. It may be considered unethical that you have used a very small number of patients, you may have exposed them to risk, and still you are not getting the results that you were desiring for. And there is something called as a statistical power, which will be also questioned at the end of the study if you do not get the desirable results. So in short, if you have a very small sample size, it will undermine the internal and the external validity of the study. To conversely, if you want to say that why not just choose a higher number, if the smaller number is making me not do the study appropriately, why not choose a sample size? I would say large sample would be 30, it could be 1000, it could be 2000. Why not just decide it to do? But each and every therapeutic area or the disease condition may not allow this. It may be countered unethical. For example, if you use a study which is for oncology, and your intervention is something which is still new and you're not aware of the risk involved into it, so you're doing this particular clinical trial, allowing the patients to undergo on that particular treatment and making their life at risk. So your ICH guideline or your ICH principles that you have, the GCP principles, are getting violated and are questionable towards the end of your study. So this will, of course, involve more finance. So since we are going to conduct a trial, it will involve finance in terms of patient visits, in terms of the treatment that has been given, and of course, all the submissions that we do to the ethical regulatory bodies, ICF creations. So all this is going to lead to a problem. And hence very large sample also tend to have something like small differences. Here the example would be that maybe you get a p-value which is very highly significant, which is like, going up from 0.00001, and this is going to say that why have you used it when you could have taken a small number of patients rather than going to the patients around 1000, 2000. So in such a situation, it will be helpful that if you can achieve the statistically significant or clinically significant results with less number of patients, we would not use a choice of a large sample size. Now, what are the important, I would say, requirements for the sample size estimations? Now, these are something that is projected on your screen are the basic requirements where you need to have type 1 error, type 2 error, and power. I've also mentioned something called as null hypothesis and alternative hypothesis, where null hypothesis is specifically designed in a way for any clinical trial where you thought that your research question should be answered. So you have to always design it in a reverse form, and you say that, for example, a drug A and drug B is being compared. So you would always try to say that so for example, your drug A is something which is being under investigation. You would say that that particular drug is not performing well as the drug B. And then you have your alternate hypothesis stating that yes, your drug A is doing better than drug B. So towards the end, when you get significant p-value, you can mention that you're rejecting that particular hypothesis and accepting the alternate hypothesis with, of course, the terms and conditions that come along with the clinical trial in terms of adverse events or in terms of any other important considerations in the clinical trial. So your type 1 error is committed, which is usually called as alpha. In other terms, in a logical sequence, it is called level of significance, and it is set to 5%. And this 5% is a number which is given to us by the regulatory bodies and, of course, from the statisticians who have investigated so far, depending on the history that we have on clinical trials. Next is the type 2 error, which is represented in a Greek letter beta, where you are committing an error, and this is something where you are accepting an intervention which may not be helpful or which may not be efficacious or safe in the patients. So in such a situation, you're committing a type 2 error, and usually this is considered to be 20%. Again, this 20% is something which is coming to you with the past experience, with the trials that was done in the past from the history, and of course the regulatory requirement. One minus of this particular type 2 error is called as power of the study. So here, when I say that power of the study is to be considered, 80% is something which is a desirable, more than 80, or 80 is something which is a desirable power of the study, to say that you are confident enough to say that the study is working well in the patients in terms of both safety and effectiveness. This is the graphical representation which you might have seen earlier as well as now, but this is something which is a very important table which tells you what is a null hypothesis, what are the different decisions, and what are the different errors that you're committing when exactly you are doing this particular type of any of the clinical trials. When you say that if you're accepting the null hypothesis and it is exactly by in fact true, indeed true, you're doing a correct assumption. And same is all for the other boxes that you can see on your screen. What could be the other requirements? As rightly mentioned by Neha in the first session, you have to design your study in a right way. So depending on your objective with the investigators, the design is been discussed with the statistician, where you know that what exactly should be the design, what should be the proportion of the design. Hypothesis testing is something which is the research question that you would answer, and it is dependent on something called as your primary objective and the study endpoint. Why primary? Because that is the main need of doing a clinical trial. In certain studies, when you want to do the comparisons, it could be in terms of expected response. It could be a study where you're doing, for example, a pain study, and you want to see if there is a relief in the pain and you're getting the response from the patients. So there is something that you're doing a comparison between the two drugs which are there. There's also something which is like the clinical important meaningful differences or the margin. This is especially important when you're conducting the studies for equivalence, non-inferiority, superiority. And of course, it could be in terms of any kind of pain relief studies. The treatment allocation, sometimes it could be most of the time when you do those studies with parallel or crossover or any of the clinical design, mostly there is, if nothing being mentioned, it is always 1:1. But there could be studies where you do not want to have patients to be at risk. So I gave you an example on oncology or any serious disease condition where you do not want to kind of put the patients completely on that. So if you have a placebo controlled trial, maybe you're conducting a phase 3 study, you also have an active comparator, you also have a placebo and multiple kind of comparators. In that situation, you might want to do a study where you have 2:1 proportion, where you would consider that maximum number of patients get the treatment rather than getting the placebo, which is just going to be a sugar pill. Any kind of planned or unplanned interim analysis. Now, why we want to consider this during sample size estimation? It could be in between of the study as well, or maybe not be pre-planned. So in such a situation, if the trial is leading to non-effectiveness or there is a lot of safety concerns raised during the study, in such a situation, there is a need that the sample size estimation has to be done in an appropriate manner right at the beginning of the study, if not that, during the interim analysis. Of course, there would be in the study, there would be patients who may withdraw themselves, some may not, some may be not to follow up. So in such a situation, dropout rate also has to be considered. And the formula that you can see on your screen is how you actually calculate the actual sample size considering the dropout rate. So if your sample is estimated for a certain number, then D is the dropout rate. So it could be in form of percentages like 10%, 20%, depending on the study. And the adjusted actual sample size is going to be derived using the formula presented on the screen. Now, what are the factors impacting the sample size of the study? So there is a beautiful drawing here which says that when the sample size you're estimating, what things you should keep in your mind. More the power, better, more the sample size. And it is vice versa also true that higher your sample size, more is the power of the study. They are directly proportional to each other. The desired significance level is also important. So the significance level, when you consider 5%, it is something which is the usual way of doing the significance level. What is the variability that you have? What is the ratio you have? And what is the effective size that you have to measure? Now, effective size is nothing but you're trying to find out from the study what particular variability is existing in the data, how much far you are from that particular estimate that you are looking for, or who is going to answer your research question. Now, how do you calculate the sample size? Of course, you have ready-made tables available. You can use the formulas that are available for calculation of the sample size. The snapshots here is presenting something which is like a ready-made table available. You can use nomograms. You can use different applications like Excel, SAS, Python, R, or these provide you with these nomograms. These nomograms help you give you a kind of range of values that at particular value, what would be the number of patients required and how much you are able to get the particular kind of. So this is just an example in front of you. And you can use these nomograms for your sample size estimations. There are some other authorized computer softwares also. These are not limited. There are many more, but these are something which are applicable and are useful, or are also kind of authenticated by the regulatory guidelines. So you can see SAS, East, R programming, PAS, Python, SPSS, Data, Enquiry. Some of them are free, like you see R, Python, and they are free of cost, while others will require a license key and you need to buy them. Now, considering this, today I'm going to share a few of the examples using either of these softwares and show you how we actually do the sample size calculations. So here I've taken a sample size example, and this is specifically when you have, say, two independent needs. This is a research question that the researchers are wishing to compare two different types of local anesthesia, and they want to see what is the difference in the time to loss of pain. So there will be two treatments. One treatment is something which is an established one, and one is under investigation. And it is known from the feasibility or the pilot studies or from the literatures that the anticipated time to loss of pain for one of the type of the anesthesia is 9 minutes. And now researchers would like to generate a sample size where you have 90% power. And now here they're also mentioning about the null hypothesis, where they say that the two differences at least 2 minutes. Means, for example, if there is 7 minutes, that means the pain is reduced in a within lesser number of time. And they are asking that can we have with 90% power and with significance level like alpha being 1% and 5%. We want to see for both the things, and we want to do the study in that way. And from the past experiment, as I said, they must have conducted some pilot studies. They know that the standard deviation can range from 1 to 5 minutes. So the variability you have to see from 1 to 5, meaning incremented by 1, like 1, 2, 3, 4, 5. Now, using this information, one can do it in SAS. The procedure is called proc power. And the statement that you're using, since it is two independent means, directly you have something called as two sample means. You're doing the difference. The group means available to you is 11 and 9. Standard deviation, they have incremented from 1 to 5 by 1. And they have asked what is the sample size, with alpha being 1% or 5%, and power is 90%. But when you present it in a form of calculation, it will be 0.9. Now, this is towards your right. If you can see the display, you can see that all the different options that we have given to this particular SAS program, there are different sample sizes available. And based on this, an investigator or the sponsor company decides that what exactly I want to do, which should be the number that I want to take. Next, here I'm giving you an example of a crossover 2x2 design, and this is specifically for a bioequivalence study, which is studying equivalence. So, since it is a crossover design, there are two treatments and there are two formulations and denoted as A and B. And what they are trying to do is they want to have the parameter of interest here is a PK parameter AUC 0 to infinity. The geometric mean is something which is provided, and as rightly said by Neha in the first presentation and also by Mahendra, that we are looking at the range from 80% to 125%. So in this study, they have given us some information from the past studies or from the literature review that what should be the sample size. And they're looking at an equal allocation, which is 1:1. And the geometric mean ratio here is 1.1, and they want to see the equivalence to be achieved. From this, they have also known, and it is important to know that which particular estimates do you need really to do this calculation is something called as variance. Now, they have a common variance and they have given you a certain number, and that is 0.1003. Now, getting all this information, you have something which is already in place, which is the GMR, which is the geometric mean ratio. They have given you 95%, meaning you are having 5% of level of significance. And that is how you're going to calculate this. Now here, this is the representation again using the proc power. Again, we are doing a two sample test. Alpha is given to you. Now, these things which are presented on top are nothing but the macro calls, meaning you're just defining what kind of number you're going to put inside the proc power. And since this is a bioequivalence study, you have to do a log transformation to the variables. And that is why when you define the variables, you're using a log function for whatever values are presented in the example. So when you say 0.8 to 1.25, you're using log to that because you want to convert the data into that tends to normal distribution. The presentation or you can see that the number that is estimated here is 68. So for 95% of power, you can see that there is 68, which is the requirement. And this with the click of buttons and using the right syntax in SAS, you are able to get the 2x2 kind of a study sample size. This is another example where the sample size is using proportions. So most of the studies, or I would say in hospitals or in the clinics, whenever there is a new test or there is a new device or anything which is a new tool which has to be used in terms of radiography and also in terms of any of the devices that are designed in order to for the brain function, or just to test the certain scans, sensitivity and specificity play a very important role. Now, why they are under proportions? Because they are usually presented in form of percentages or in form of simple decimals. Now, this is the study where they are saying that for a certain type of a cancer, the sensitivity and the specificity is known, which is 71 and 82 respectively. And now they have a new diagnostic test where they are trying to see that whether that particular test is going to be a little less expensive and, of course, invasive. So this is specifically for a study, like oncology, where there's a lot of invasive procedures. It is very expensive. So in such a situation, they are developing a new diagnostic test, and they want to see the credibility or the kind of authenticity of that particular tool. They've given you that what are the different expectations out of it, like what could be the different sensitivity differences that they're looking at. And also the prevalence is important because they want to see what could be the population size that they can see, draw the sample from, and it is known to be 6%. Now this could be specifically to that particular institute where they are doing the study, or it could be for that particular region. And the required thing is that the power that they are expecting, they're looking at the sample sizes incremented by 300 from 300 to 3000 with the, and they want to consider 10% increase in specificity, which means like 90%. So using this all information, one can use a software. Now this presentation in front of you is from a software called as PASS Pass. And of course, this is something which will require a license, and it is a paid software. So we could do it using formula as well, but considering the time constraint that we usually have in a way that the softwares are developed for us to understand that how fast we can do this and get to a study to be started. So we are solving for power, as they have said. We are doing the sensitivity and specificity both. The alpha is the 95%, so sorry, the 5%. And the sample size they have mentioned to be incremented, which you can see on the screen. And the prevalence is 6%. Whatever is the null hypothesis that is being presented here for both sensitivity and specificity. So with the click of button, you can get all the different options in the pass, and this is how you can generate the sample size easily using pass software. Another example is on a paper that I got from Annals of Oncology, Medscape from 2012. And what I have done is I've taken the snapshots of that particular paper and what is the assumption that they had. And I have recalculated that using a software to understand that are we able to achieve that what was done in the past has been rightly done to achieve the research question that they have mentioned in this particular paper. So here, this is about the presentation that is done in the paper, and I'm just giving you a snapshot of what they presented in the sample size section in that particular article. They have mentioned here that they require at least 24 subjects or the cases in either of EGFR positive and negative cases. So totally 48 patients are something which are required. Although they did not mention the software or the way of calculation that they have done in terms of what software has been used, I have used the same logic what they have used here, and I have presented something. If you look at the prior this thing, they have mentioned about the average half width. Now, what is this particular terminology usually used? So a slide presentation here, like when you have confidence limit, you have an upper and a lower limit. So right from your point estimate, which is the mid value, towards the either of the limit is the distance, which is called as the half width of a of the confidence interval. So after presenting these values, I've got something here as a sample size. Now this particular snapshot below, which you can see, is from nQuery Advisor. Again, this is a software and it is a paid software. Although you can use the formulas, but here something is very pretty much faster. You can use this and you can see that the sample size that I got the estimated is 25. There could be a rounding error, and that is maybe in terms of confidence interval, in terms of proportions that we are presenting, or in terms of one-sided, two-sided test. So in that way, I've got at least say 2 patients higher to what was actually projected in that particular paper. So here in the prior example, what I presented is 24 is something which is a sample size per particular point, and here it is 25. This is another research paper which is talking about non-inferiority for a crossover design. So I'm giving you the source. This is a recent paper from 2022 where we have efficacy and safety of meteoritic microdrops for a retinopathy for prematurity screening study protocol for a non-inferiority crossover randomized control trial. So, and next is the snapshot of what exactly was presented in the paper is projected on your screen. Here they have used R software, and the package that they have used is PowerToast. The required data that they have is like power of 90%. They are having one-sided significance of 2.5%. What is the mean difference that they have mentioned? So there is a nice entry made in the paper about how they have done the sample size estimation. Now, since they had used our package, I'm trying to reiterate and calculate that in another software and see if I'm getting the sample size as they have presented here is 74 patients. They've also assumed a 20% iteration rate. So I'm using here Enquiry Advisor. The required data I have put in, where they have given us what is the non-inferiority margin, what is the level of significance, what is the expected difference that they are looking for, what kind of data this is, like this is for crossover design, what is the standard deviation, what is the power expected, and you can see that sample size per sequence that is expected is 37. So by default, it is 1:1, so 74 subjects that we have got. So here you can see that non-inferiority margin was some question that was asked earlier as well. So this margin is something which is not just decided by the statistician or by the investigators, but this is dependent on any feasibility study, past data, review of literatures, or anything that they must have recently collected and seen. So that is how we decide on the non-inferiority margin. This is another example, again for non-inferiority for survival data. So here the researcher is planning for a non-inferiority trial using a parallel two group equal sample allocation. And the non-inferiority hazard ratio is said to be 1.2, the power of 90%, significance level of which is 0.025, and which is 2.5%. And they've given you the hazard ratio should range between 0.5 to 1, incremented by 1. They're looking for a probability of having an event of 50% in the control group and 30 in treatment group. So there's a lot of things that they give you. Depending on your research question, you have to decide what formula, which software, and what kind of methodology has to be utilized in order to get it. If it is not available, you have to go and search for it, or you have to assume certain things depending on your experience in that particular disease condition or the patient population. So again here, this is an enquiry snapshot. Since they are incrementing it from 0.5 to 1, the expected hazard ratio, you can just enter these values, and within click of buttons, you can get what should be the sample size which you need, or what should be the number of events that you need. Here, I just want to make a point that sometimes it is not always just to estimate the sample size. You can do a back calculation where you can say that I want to see what particular sample size can achieve that particular power, or what should be that particular significance level that it can achieve. So you can say that you consider a sample of say 1,000 patients and then go back and see, is it giving you enough power of 80% and above to get the correct results for your study. Last but not the least, this is an example for a phase 4 for a phase 4 study, which is for a cohort study. Now, this is another paper, which is again a post-marketing surveillance of safety and effectiveness of nantimetine in the management of opioid-induced constipation in patients with cancer pain in Japan. And they have given you like what is there in the sample size section, what and how they have calculated. Again, I have used a software here. I've used what are the significance levels. So what we do as a statistician, take out the estimates or take out the required information from the papers and then decide, are we getting the right sample size for our study to be conducted. So, and I've given you the sources as well where you can go and check out this particular papers, and you can also do the recalculation using a different software or the formulas. So the key takeaways from my presentation would be that whenever we do any pilot style studies, there we need not do a sample size calculation because they are just pilot studies. It's only that whenever you give an interventions, it has to be considered. And sample size is something which is not just any number that you decide. There's a lot of research behind it where you do literature review, you have in line with the investigators what kind of population you are doing, what study design it is, what is the difference of association or the association you are studying, who is responsible for it. It is not only the statistician but each and every stakeholder who is conducting the clinical trial. And of course, you're using the right methodology in order to do the sample size estimation and the software. So thank you all for this, and the session is open for the questions.
All right. Very good. Thank you, Aarti. I think most of the question I tried to answer on the chat, and not the chat, the question answer. Some of the questions mentioned here in the chat as well. SAS only easy to calculate. You can use SAS, you can use R, you can use Python, whichever you are comfortable with. So all the softwares are using the standard methods that how we calculate those sample size. So there is no preference for one software over other, but try to use the validated software. Yeah.
Yeah.
So that's important. Very good. Then some question here. How to conclude the dropout rate as based on the past study dropout subject may vary, then how to select dropout rate. Dropout rate you assume, yeah, either based on the previous literature you say dropout rate maybe 10%. If you have doubt, because clinical trial your role will not start once trial is over, your role starts when trial is starting, and over the time you look for the data from your clinical operation and data management team about the dropout rate in the study. So that will help you to understand whether dropout rate what you assumed and what the actual, are they too different, then you need to do something if they are aligned. Because if a long duration study, maybe your dropout rate could be higher. If it's a shorter trial, your dropout rate could be lower. If your patient population is elderly population, maybe dropout rate could be different than the general population. So it has a lot of things to consider while you consider the dropout rate. Again, it's not only you as a statistician to decide, you take insight or input from your clinical team as well. Okay. So basically use a software calculate sample size in two way and multiply it by 0.5, and for some replicate multiply it by 0.75. Okay, very nice question. So when you have random allocation of 1:1, then you multiply your sample size by 2. Okay, if you are comparing two means, and if your random allocation is changes, instead of 1:1 it becomes say 2:1, then your ratio is no longer the same, and that your multiplication factor is also getting changed, and this is where sample size also get reduced. Okay, so you need to consider that which option you are looking. Okay, so that's how the software also considers. Software name, as I Aarti mentioned couple of things. You could use and query is a all license software. You can use pass, you can use east, you can use SAS, you can use R, you can use Python. There are so many softwares available. There are Excel sheets also created like a software, you can use it, but those Excel files do have inherent macros, and some organization, or again, I would not prefer to use a micro in built macros in a Excel file, because that could have potential risk as well. So please avoid using those Excel files. You can use mathematical calculation, you can use pen and paper. So there are so many ways to do so. All right, I could see some question again popping on the question and answer. uh, Whether crossover studies concluded only for bioequivalence, what else you would like to conclude from a crossover study? It depends, why you are designing crossover study and what you want to conclude, but mainly these type of study designs are used in bioequivalence only. uh, Sample size of R and do not match the highly variable drug in bio. See, a highly variable drug means what? How you decide whether drug is a highly variable? You consider the variation or the variability, and then you decide whether drug is highly variable or not. And then your criteria will also change, because then it's no longer 0.8 to 1.23. Then the criteria might also change, little different. Then you say how to justify the sample size for FDA when the pilot study data is not reliable and reference data not available. Then you need to do something, because if nothing is reliable, then you need to come up with some assumptions, and you need to justify that why you are making those assumptions and how you test those assumptions over the conduct of the study. You start with 100 subjects, and then you say based on 100 subject, maybe I will try to estimate the variability, or maybe I'll try to estimate the difference what we would like to see, and then again you can re-estimate. So there are statistical methods, but every such thing have advantage and disadvantage, so you need to take this decision wisely. When calculating sample size or a facial cleanser study. Okay, this is I think some study related to cosmetics. Should I focus solely in on article about facial cleanser, or should I also consider studies on related product like face wash, or I what you want to compare and what you want to claim, and based on that you decide which one you would like to consider the data. If you have three different things, and if you're not sure which one to use, maybe you can, as a statistician, you can consider, okay, maybe I'll take the average of these three, and that I can use. Our software is valid as this type of open software is raised data integrity. Software is valid because there are packages for R to calculate the sample size. Again, the packages are developed by some individuals and they are not licensed, they are not paid, so no one will take guarantee whether something is wrong in those packages. You need to use those wisely. Then it says the sample size vary from regulatory to regulatory, for example FDA, EMA, or for all the regulatory sample size. I think the study when you conduct the study for one study or one trial protocol, and in that you justify the sample size. Maybe that's protocol you would like to submit to FDA or EMA or some other in agencies. By agency you will not change the sample size. Okay, agency might ask you to add something more in terms of the clinical perspective, but sample size is not really varying based on the agency's requirement. Yeah. If so, there must be a reason why they are asking you to change the sample size. If we calculate sample size in SAS and R, it gives different result for the same parameter. For that, I have searched for one research paper which the author was in the US base. He suggested that we should prefer R for these conditions. Why? Can you please explain? Okay, so I don't know whether what article or paper you are referring. So if you have anything to add, Aarti, here.
Yeah. So here what happens is both the packages have a different way of calculation in their search engine. So R uses different packages. So like when you talk about count data, R uses something which is RPact. SAS has
Mm-hmm.
something which is commonly used across different kind of endpoints. So, like if I talk about this two sample means which I was talking in, it can be used for multiple things. It all depends on your regulatory body as well. Like when you're submitting this, if I consider still in India, I would not see that everybody who is submitting for R is getting, the non-query, like they are not getting any query from the regulators. So it is always easy to use R because there is a package and you just need to enter the data. But the same is the case for SAS.
Okay. Very good. I have one question for Aarti ma'am. For clinical trial, how to calculate sample size? In BAB, we need CV power, B limit is enough to calculate sample size, but for clinical trial, can you explain detail like what are the parameters to be used?
So again, it is dependent on your research question, like what is your primary endpoint. Once you look at that, you will need certain assumptions. If you do not have any past data to look at, or you do not have, feasibility or pilot studies to conduct, so you have to assume things. uh, of course, alpha, beta, and some and the power of the study is common throughout. However, when you're looking at CV, this is something which is in the generalized, a different region study. It is not always for a certain kind of an area or a region, but it is multiple regions which is done, and that is why when you use CV over there, you can use that. However, everywhere it is something where you talk in terms of variability. So different population or a different region will have different assumptions which you need to calculate for that particular region and for that particular patient population.
Yes. Very good. And then when you calculate sample size for clinical trial, maybe you don't need CV, but what you need is the clinical meaningful difference that you would like to detect. And then also you need the variability, if or the
Yes.
if you get from your clinician the effect size, you don't need both. Then you need only the effect size, because the effect size is the ratio of your clinical meaningful difference over the variability. So you can use effect size instead of the mean and the standard deviation. You need power, again you need the significance level. So again, if this is when you have endpoint based on the means, and if you or change from baseline, if you have some different type of the things, maybe if you have time to event, then you need different information, then you need hazard rate and so on. So based on the endpoint, you decide what information is needed for that. So how you analyze that particular endpoint, try to use the similar methods for the sample size calculation. Okay. Which software is easy to calculate sample size? It depends. If you are very comfortable with R, then R is easy. For me, I like R, I don't like SAS because I don't know how to use SAS. Which study design lead to lower sample size, non-inferiority or equivalence, and why? Now the formula, if you look at, so I'll give you one example of change from baseline, and we would like to see the difference in two treatment groups on certain parameter. Say blood pressure, very easy to understand. So change from baseline in blood pressure in treatment A versus treatment B. Now when you the formula says in the denominator, whatever the difference that you would like to detect will come in the denominator. Now if you have the superiority type of study, it means you say the difference is zero, that's your null hypothesis, right? Then your clinical difference will remain whatever you assumed. Now you consider the non-inferiority type of study. In non-inferiority type of study, what will happen? In non-inferiority type of study, you have the non-inferiority margin as well, right? So the non-inferiority margin will also get subtracted from the difference what you have, and then it becomes even more less, and that will different of that will change your sample size. Random allocation could also influence your sample size, 1:1 versus 2:1. So there are so many things. I'll show you one example with all these basic assumptions, how that looks like. What the minimum sample size is required for bioequivalence study for regulatory submission. There is no fixed number. Most of the time, based on experience, people say 22, 24 are the number used for bioequivalence study. But those numbers again depends on the assumptions, right? So if you put your variability, you have your ants, you have t statistics value, and try to calculate the 90% confidence interval, and you see when your sample size is too low, maybe your ranges will be very wide, and you will fall outside 0.8 to 1.25. So that also you need to look into that. uh, what minimum, then I have one question for Aarti ma'am. I think clinical trial to calculate sample size, we need CV power. I think we discussed this.
Yeah.
uh, At the start of study, loss to follow-up or dropout rate is assumed to be 10%, for example, and based on that, sample size would be calculated. But during the course of study, if the loss to follow-up changes, should investigators re-evaluate their sample size? Okay, so if the if the dropout is too high, you assume 10%, and if your dropout is 30%, what will happen? You will not have enough subject at the end to evaluate the endpoint, and you might have lower power. So you need to adjust that to conclude with enough power. So maybe you might want to consider that and revise your sample size things, because you are not changing any other assumptions. Yeah, but higher dropout is also a question that
Yeah. So, Maidra, I want to add here,
yeah.
like when you write the sample size section in your protocol, you mention the minimum number of samples required for your primary endpoint to be achieved. And over top of that, then you have dropout rate. So, for example, if you're saying I need 20 subjects overall, and on top of that, again, I have 20%, the dropout rate added to it. So, I know that at least 20 patients I would need for my study to be conducted. So even if your dropout rate is increasing, you know that 20 is the number that you need to achieve in order to achieve the effectiveness and safety criteria in your study trial.
Yep, very good. Thank you, Aarti. So sample size is the least what you calculate. The minimum required sample size. It's not the maximum.
Yes.
Well said. I come across with one different scenario in one my project in case where the sample size used in efficacy result is challenged, such as claim team or court. What is such as claim team or court? How can we justify the validity of our finding and demonstrate that the sample size which we calculated or used in our study was appropriate for proving efficacy? This is very
You have to
difficult.
define your assumptions clearly, like what exactly you have used in achieving that particular sample size. If that is clear, I think the only thing is that the query has come to you and you can get the resolutions towards this. But in case you are not able to prove why you have achieved that number, definitely it is going to be kind of a question and can be challenged.
Yeah. And why someone has to go to court for justifying the sample size? Maybe regulatory agency is good enough to handle this. Okay. So once you calculated the sample size, and if you met your objective, then regulatory agency will look into that, and not necessarily that it will have that you need to go to court for justifying the sample size. Okay. If your drug is really working, it is working, whatever the assumption you made. If it's not working, it is not working. Can SAS be used for fully replicate or are used? You can use whatever software you are comfortable with. A small sample size and non-norm data, then adjust. Would you suggest to ensure accuracy? How would you estimate sample size in R for a study with a small? I think this is requires a separate session on R,
Uh,
that how you do this calculation. But your question is well taken. We'll try to address this in some other sessions. Can we have alternate software for calculating statistical analysis rather than R and SAS? Because SAS and R are regulatory submission software, but we have any other software particular or licensed software. If you are ready to license the software, there are many. You can use nQuery, you can use East, very expensive softwares, and R and Python are freeware, so you can use those as well. And don't think R you need any license and so on, because most of the things are available freely. But if you want to use for commercial purpose, you can also get the license through R, because then you get everything validated. What is significance of p-value? Useless. Okay, p-value is just a number that will tell you whether your study is meet that null hypothesis or not. But along with p-value, you also need to look at the 95% confidence interval, because that will also provide you a lot more information. For example, if you are running a non-inferiority study, p-value is again useless. So don't give too much importance to p-value. You need to see the estimate, you need to see the confidence interval, and by looking at those two numbers, you can tell whether p-value is less than 0.05 or is greater than 0.05. Perfect. Is there any thumb rule for percent difference among treatment of 3 or more when we make 3 or more allocation ratio? No.
There is no.
What is minimum sample size for fully replicated bioequivalence study acceptable by agency? Please do refer to the bioequivalence guideline from regulatory agency. It will provide more and more information on different topics. So you have almost all the things listed there. Is it acceptable by regulatory if sponsor makes sample size estimation adjustment in trial even when it is not planned? You know, you need
They
to
would
get buy-in
recommend.
from authority, and you can do the unplanned, because we cannot predict for future for everything. You know, when you say this is the predicted value, but in prediction in future you also have so many other covariates that could influence so many other things, right? So it is allowed. Sample size estimation, re-estimation, you need to show to agency that your type 1 error is not inflated, so that you need to prove, and you need to provide the data to agency, and they will consider that. Power of pre-study and post-study changes with percent CV or T by I or T by R ratio. Why? Power of a pre-study and post-study. What is pre-study and post-study? So maybe they calculated with some power, and when they look at the actual CV and TR, that power will change. It will change because the power calculated based on these two parameters. So definitely it will have impact. Yeah. If your T by R ratio is going way far from 1, there are highly likely that you go outside the bioequivalence limits. Is it minimum required sample size, or precisely can it be optimal? No, I think Aarti mentioned it's a minimum required.
Yes.
Hello. As we know, when study commence, calculate report sample size in the protocol paper. In this part, we assume certain dropout rate. Now, if it, I think we already discussed about this. Power of pre, I think this also we discussed. Is there any thumb rule that is also be
Have been discussed.
discussed? I think most of the questions are addressed here on this. And let me see if there is anything else.
We covered Mahendra.
Participant, okay, let me try. I could see 3 hands up. Sucheta Patnaik. Let me see if she could talk. You have only 2 minutes. We will consider only 2 minutes because we are running late for the next presenter. I think I'm asked to unmute. I think this facility we never tried. But Sucheta, Dipan, Josita, Khada, Babu, if you have any question, please put on the chat. We'll try to answer. Or Sucheta, if you want to speak, you can unmute yourself and try to speak. No, it's not working. So if it's not working, then the next presenter I will invite Swan. Swan, now you could share your screen and take us through your session.
Thank you. Am I audible?
Yes.
Thanks. And you can see my slides.
Yes,
Can you-
Subhan, go ahead.
Yeah,
Yes,
thanks.
Subhan, go ahead.
Thank you, Neha and Aarti, for setting the stage. So, hi everyone, my name is Shuvan Mondal, and today I will be presenting a practical approach towards the sample size calculations. Before we jump in, just a note that any of the views that are expressed today are solely of mine, and it doesn't really reflect the views of the organization that I represent. Okay, so agenda for today will look like, first I will uh, give a simple article from regulatory requirement, and I will not go into the details. And then the rest of the section will look like there are 3 different endpoints, and for each of these endpoints, first I will give an example, then we will discuss the theory a little bit, and then we will see the results. And for each of these types of endpoints, continuous, stress of proportions, and time to event, we will follow the same logic, and in the end, I will end with a discussion on group sequential design. So yeah, let's jump into the presentation. Uh, according to very old FDA guidance, you can see on the slide, this is actually the quotation from the same guidance, is that they need at least two adequate and well-controlled clinical trials to have any new drug to be approved in the market. Now, in this case, there are many new uh, there are many new updates to this guidance, and essentially which falls in. Sorry for that. The other mic is not working properly. Can you still hear well?
Yeah, some background noise. Maybe you keep your mouthpiece little closer.
Yeah.
Yeah, now better.
Okay. Thanks. Um, okay, so according to FDA guidance 1988, it is required to have at least two adequate and well-controlled studies. Now, as I was mentioning, there are many updates to that where only one very good, well-controlled study can get you the approval of the drug. But here, the more important part is adequate and well-controlled. Now, sample size calculation plays an important part in there because if you do not have the sufficient subjects in your study, the results are not very accurate and reliable for the assessment by FDA. Okay, with that, I will not go into the details about why sample size is very important. There are many articles in the internet, and there have been few discussions by RT as well on this important part. So I will jump into the next section, which is the continuous endpoints. So as I was mentioning, I will first state the example, and then we will go into the method and the results. So this example is actually taken from a study sponsored by Novartis. The link to this study, the city.gov link is here. And I'm mentioning it is almost to that because this example is much dialed down version of the actual study. So the study goes like this. Uh, we recruit 1-to-1 randomization ratio treatment and control patients. And for each of the patients, we follow them up for 12 weeks. At the baseline or at the start of the trial, we ask each of these patients how much pain they are getting, and that is a study of neuropathic pain. And this can be marked from a scale 0 to 10, 0 being no pain and 10 representing maximum imaginable pain. And we do the same thing for treatment patients as well. And after 12 weeks, we ask the same question. So for each patient, we have one baseline observation of how much the pain was. And after 12 weeks, we also have one observation for how much the pain was after 12 weeks after getting the drug. Now, half of them are getting placebo and half of them are getting the treatment. Now the test is essentially whether the treatment arm is significantly better uh, than the control arm in, well, controlling the pain. Now here, the endpoints are 0 to 10, all the integers. It might seem a little bit discrete, but what we are doing in the study, actually, we are taking the mean over 1 week to get each of these observations. That's why we can essentially assume continuous endpoint for this example. And the corresponding assumptions about sigma, that is the standard deviation for control arm and the treatment arm observations, can be gotten from from the past studies and all other literatures of similar kind of drugs as well. Now, for the continuous endpoints, we will discuss a little bit of theory here. So let's say xi's are coming from a normal distribution. In general, when we do not have much reasons to believe that this is non-normal, we generally assume normal because there are many asymptotic assumptions and distributions that can be assumed for normal distribution, and this is easy to calculate. So let's say the xi's represent the observations from the treatment arm and yi's represent the same from the control arm. And we know the sigma 1 and sigma 2, which is the standard deviation for treatment and control arm. And then the sample size calculation or the sample size of the total study looks like this. Now you can see the Z alpha and Z beta are essentially the quantiles from the normal distribution, and it depends on sigma 1, sigma 2. And there is one more parameter K, which is the randomization ratio. And the randomization ratio is embedded into the formula. Now, one quick question is how randomization ratio essentially impacts the sample size. Now, in our case, it was 1 to 1. So that's why we might do with the minimum sample size required for the whole study. But when the randomization ratio changes from 1 to 1 to 2 to 1 or 3 to 1, it affects the sample size required. So the next slide, we will look into one example. So here I will first explain the assumptions for the parameters. So delta is the true difference that we are assuming. Sigma 1 and sigma 2 are, well, the standard deviations for the observations of treatment arm and control arm. I just tweaked the numbers a little bit. In actual examples, the number were a little bit different. And the target power for the study that I'm talking about was 90%. So we want to design a study to achieve 90% power. So how to see the plot here is on the x-axis we have the randomization ratios, and for each of these randomization ratios and assuming all these parameters, we are saying how much power is needed to get, sorry, how much sample size is required for the whole study to get 90% power. So just for the illustration, uh, here are three colors as well with alpha 0.025, which is 2.5%, 0.05, which is 5%, and 10%. In the actual design, it was 5%. But for the illustration, I will explain one point. Let's say this one. So we have these assumptions, delta equals to 1, sigma 1 and sigma 2 are 1 and 2 respectively, and we need a power of 90%. Then what we say is at 3 to 1 randomization ratio, uh, to achieve 90% power at, which is red, so we are at 5% type 1 error, we need a total sample size of close to 150 parents for the study. Now, this is power was fixed for this, and in many trials, when we do not really uh, well have an idea of how much sample size we require, because there is always this back and forth between how much sample size is required and whether the assumptions of the sigma 1, sigma 2 and delta are true. So thereby sometimes we also generate sample size for different power criteria as well. So in the next slide we will vary the power. So here we are fixing the randomization ratio to 1 to 1. Now on the x-axis we have the power. So previously we had 90% power, and now we will see whether for 90%, whether for other scenarios, what kind of sample size are getting impacted by the power criteria or the target power. So with the same assumptions here, delta, sigma 1 and sigma 2, and randomization ratio to be 1 to 1. Now, if we try to explain, let's say this red point now. So it says with these assumptions, to get 95% of power at 5% type 1 error, we need, which is close to 110 patients for the whole for the whole study. And this is essentially treatment plus control, total patients. And here the randomization ratio is 1 to 1, so it is 55 each for each of these arms. And that's how the sample size is getting calculated. Here I want to set out one vocabulary for the audience, is that these sample sizes are getting calculated by a closed form formula, as you have seen in the previous slide, this one. And when you have a closed form for the formula for the sample size, we call the sample size calculation to be coming as a nominal sample size or calculated as an analytical approach. But there are many scenarios when we, let's say, we do not have the normality, or for other distributional assumptions, we do not have these closed forms. Then we use some other methods, and I will explain them in the later slides, and we call them the empirical power, which is essentially we are generating some data and calculating as a proportion the power. Okay, so after continuous endpoints, we jump to endpoints which look like proportions, uh, which can be as simple as you have a coin, you toss it 100 times, and you are trying to guess the p of it, whether it is half or as it has the chance of head and tail equal or it is not. But in clinical trials, I will give you one example from the clinical trial setting. There is one book from which I mean, these examples are inspired. I refer to this book in the reference section later. But to explain the example, It is again a two-arm trial, treatment, one test compound and one control arm. So for the treatment arm, uh, the P2 or the proportion of the event. So it is essentially, let's say the patients are starting sick, and what is the probability of each patient of getting better? And that can only, I mean, that can only have two types of responses: whether the patient got better, that means one, or the patient didn't get better, which is zero. Now, by P2 equals to 35%, we are saying that we are assuming the treatment compound that we have, the test compound, will have a probability of 35% of getting better for the patients who are getting this drug. And for the control arm, let's say they are getting placebo, they still have a probability of getting better to 20%. Now, we want to test, or we want to design a study whether, with these assumptions, how big the study should be so that we can produce 80% power at 5% level of significance or 5% type 1 error. So let's see a little bit of theory. I will not go into the details of this theory, but uh, our null assumption here is that the test drug doesn't work. That means the test drug proportion of getting better is equal to that of control drug. And on the alternative hypothesis, we test this is not true. Now, in many cases, there are notions of one-sided test and two-sided tests. Now, this is an example of two-sided test where test can be worse than the control as well. But in our example that I will be showing the calculation of in the later slide, we will be using one-sided hypothesis. And in that case, we can generate and we can write the hypothesis as P1 less than P2. That just depends on how you want to spend the alpha on this hypothesis. Now, for this type of examples, there are two approaches. One is when we see the differences are small between treatment and control, and we are trying to design a bigger study, then we can essentially assume the asymptotic distributions and essentially large sample calculations, which are easy. Now here, what I'm going to show is when you have a smaller study to design, then those asymptotic assumptions do not follow, and we generally use the Fisher's exact test. Now, just to speak loosely, when we have the assumption of how many subjects are getting recruited, the distribution of how many subjects from the treatment arm are getting better, that can be shown to be following a hypergeometric distribution. Now, I will not go into the details of this distribution, but what I want to mention here is the calculation of this p-values or the probabilities do not have a closed form. And what you have to do is you always have to add up the extreme probabilities to get the p-value for each of these studies. Now, there is a simple way to calculate sample size for this kind of studies, for a smaller study or where you do not have the closed form for the p-value. And that is one device I am going to explain in this slide. So we know the P1 and P2 as mentioned in the example part. And what we will do is we will simulate or generate thousands of similar looking studies. And for each of the studies, we will compute the p-values. And at the end, we will see for how many of this, let's say we have designed 100 similar studies, and we were able to reject the null hypothesis in, well, 80 of them. So in more than 80% of the studies, we were able to essentially reject the null hypothesis, and that is the candidate value of our power. So what we do is we have the similar assumptions of P1 equals to 0.2 and P2 equals to 0.35. So we generate, let's say we are assuming 1-to-1 randomization ratio here. So we generate n values from treatment arm with binomial distribution. This can be done easily in any of the softwares. I used R for all the calculations. So let's say we generate n values from binomial distribution with the p, that is 0.35 for treatment arm, and with the p of 0.2 for the control arm. And now that we have n values from treatment and n values for control, we use this test, the Fisher exact test that I was mentioning, and from each of this test, we get one p-value. So we have 100 studies. I have I have calculated 1,000 for more accuracy, but for example, if you have 100 studies and you generate these 100 p-values, and your empirical power, we call it empirical again to clarify, is that that is coming from a simulation process, or essentially coming from the data that you generate. So your empirical power is for how many studies or for how many replication of the similar design you were able to reject the null hypothesis. And looking at the result, I will again explain the construct here. We have the same P1, P2, and we are using randomization ratio of 1 to 1. Now here, on the x-axis, we give the sample size per group. That means the total sample size is double of this number. And on the y-axis now, we have the power. Now in this simulation cases, we do not have a closed form, so we don't really get only the number for which we get 80% power. But what we do is, for a range of sample size scenarios, we generate the empirical power and see for which minimum number of sample size it crosses the 80% bar here. Now, just to go with our own example, we needed 80% power for our study, and we needed 5% error, which is the red curve. Now, this red curve processed the 80% bar somewhere after 120, but before 130. So close to 125 patients per arm. And obviously, there have been discussions about how to adjust for the dropout rates. You can assume something, and that assumption has to come from the uh, well, from the recruitment agencies or from the people who are well responsible for the recruitment and also the clinical. There are many reasons for this dropouts, and this can be adjusted on top of this number. So 125 good proper observations that you need. Now, if you think if you recruit 200 patients and then only you will get proper observations for 125 patients, then you have to adjust that way. But this is how the basic sample size calculation goes for the proportion endpoints, but there are large sample counterparts as well, which are easier than this to do it. With that, I will move to the next section, which is time to event. Now, time to event is different from the continuous endpoint in that sense that, let's say we are dealing with lung cancer patients now. Now, if we start with 200 patients who are clearly at the late stage of their disease, during the trial, many patients might die or may drop out from the study because of lack of efficacy. But after 2 or 3 years, there will be many patients who will not drop out or can still continue. Now, in those cases, there are some observations you have because the time to death is observed. But for some patients, this is not observed. Let's say after 2 years, there are 20 patients who are still ongoing. Now, for those patients, only information we have is their time to death is more than 2 years, but we don't know anything else. Now, in those cases, we cannot treat this data same as the continuous data that we discussed in the previous slide. So there is a different approach to deal with time to event. Now, I will give you one easy example, which is again taken from a book I referred in the reference section. So let's say we have one-to-one randomization and we have two drugs. One is the test drug that we have, and other is the control arm. And this was alluded to in the previous talks as well, that in cancer trials, the disease is so deadly, it is unethical to just give them sugar pills or placebo. So in general, we give them the standard of care that we have in the market, and we try to prove ourselves better than the corresponding standard of care. So in this case, the control is chemotherapy, and we are trying to test whether our drug does better than the the chemotherapy. And we follow up each patient for 12 months. Uh, Now, there are some assumptions that we generally get for the taste drug from phase 1 studies or from molecules that have kind of similar composition of the taste drug or any previous trial of similar kind. So for our case, The 6 months is the median survival for the chemotherapy. So o- half of the patients will die within 6 months if they are taking chemotherapy. This is I mean, how you can loosely understand what median survival is. And 9 months is the median survival for the test drug. Now you can see we are assuming the test drug to be better and trying to generate or trying to design a study which can give us a 90% power at 5% type 1 error. Now let's go to the theory section a little bit. I will not go into the parametric distributions, but what I want to mention is there are many distributions that you can assume for this time to event. The easiest one is exponential because it has constant hazard throughout the time, but there are Weibull Gompertz as well, which are well, I would say a little bit tricky to calculate. Now for exponential distributions, for calculating a closed form for the sample size, uh, well, the calculations are a little bit involved in terms of double integrations and everything, but the formula can be found in the book that I will refer in the reference section. I will show you an easier version of rather than using the formula and how you can essentially calculate the sample size for a time to event. Now, just want to mention that our assumptions were treatment has 9 months of median survival and the control has 6 months of median survival, and our target power is 90% and 5% is the type 1 error that we are looking for. Now this has the similar structure because this is again the simulation framework with which we will calculate the sample size. Now here we will take the easy example so that we are assuming that the time to event are generated from exponentially distributed, are generated from exponential distribution. Now, the rate of this exponential distribution can be calculated from uh, the median survival assumptions. I mean, there are many studies where people assume the hazard rates, and there are many other studies where people assume the median survival. But for exponentially distributed data, these are one to one. So if you know the median survival, then if you know the hazard rate, then the median survival is log 2 by the hazard rate. And if you know the otherwise, that is, you know the median survival, then log 2 by median survival is your hazard rate. Okay, so having said that, how to calculate the hazard rate for each of this distribution, we do again the same thing. Let's say we have this 1-to-1 randomization ratio again. So we generate n patients from treatment arm, and then we generate n patients from control arm using exponential distribution. And when we are generating from treatment arm, we use the corresponding hazard rate for the treatment arm, and for the control arm, we use the corresponding hazard rate for control arm. Now, we do this, let's say 100 times, and for each of these studies, we will generate the p-value and at the end, we will see how many of them are essentially lower than this alpha. In our case, this is 5%. So in the next slide, I will show you the results from 1,000 similar studies and getting the proportion of how many times the null hypothesis was rejected, and we call it the empirical power. This is similar to the proportion case. We again have the sample size per group on the x-axis and the empirical power on the y-axis. Now, here I note down the assumptions. We have median survival for the chemo at 6 months, median survival for the test as 9 months. We use randomization ratio of 1-to-1. And let's say for our case, we needed 90% power, so we use this, and 5%, so we use the red color. And we can see close to 120. It is slightly more than 120, but yeah, it's close to 120 subjects per arm. That is 240 total, gives you the power of 90%. So this is a safe number with which you can design a study. And of course, there is this adjustments for dropout rates and everything. And these days, the study designs are very complicated to essentially calculate, and that's why we generally prefer this simulations than the formula, because you assume some dropout rates, you assume some other censoring rates as well. So this makes it very complicated. That's why simulation comes into the picture. But for a simplistic study design, you can calculate the sample size like this. Now, these are the three types of endpoints that I have discussed so far, and I will just finish my presentation with a short discussion on group sequential methods. These days, what happens is, let's say you have designed a study and your follow-up period is, let's say, more than 2 years. Now, there are many adverse events that can happen to patients, thereby, I mean, you need to essentially terminate the study. And there can be other good things that can happen to your study, so that you assumed, let's say, 0.15 as your true treatment difference, but your treatment is much better than your assumption. So what can happen is, by first year of your overall 2 years of assumption, the data is so good that you do not want to spend any more money, and you want to, well, just report this data to FDA for approval. So for those cases, you cannot really abruptly stop your study and look at this data. You can essentially keep a window of looking into the data in between the trial, and you can mention earlier on while designing that if my data is too bad, then I will just stop my trial here. But if my data is very good, then I will stop my trial as well and report this data to you for approval. So I will not go into the study for another 1 year after one. So group sequential methods, which essentially tells you that you have a 2 year study, but after, let's say, half of the patients are recruited, you want to have a look at it. So when you have when you are trying to look at the data in the middle of the study, let's say after 1 year, then the boundary, or let's say these gives you implications on the p-value or the test statistic. If this goes beyond these boundaries, then only you are able to stop your study for efficacy. That is overwhelming efficacy, and you want to report this trial. There are many historical bounds that are used. This is Pocock's test, O'Brien's Fleming. I am not explaining the designs and alpha spending functions here, but I will show you a quick example with Pocock and O'Brien Fleming. So let's take one example of a binary response, and this is we have lesser difference between P1, P2. We have reasons to assume that this study can get bigger, and in this case, we have assumed the asymptotic assumptions. And if we have a fixed sample design, so that we don't want to look into the data in between, we will follow them up for 2 years, and only at the end of the study, we'll look into this. In those cases, let's say, I mean, the calculation shows that it is 385. The calculation can be similar to the simulation structure or the framework that I have shown before. Now, if we now want to look at the study, look at the results 4 times in between. So this k equals to 5 is essentially calculating the last look at the data as well. So after 2 years, if we look only once, that means k equals to 1. If we look into the data total of 5 times, 4 times in between, and 1 time at last, we assume k equals to 5. And in those cases, the Pocock boundary inflates the sample size up to 474 from 385, which essentially means that if you want, the more times you want to look at the data, the more sample size that you required for the study. One thing I want to highlight is, between Pocock and O'Brien Fleming, Pocock is now a little bit obsolete because it gives a constant decision boundary for your trials, and which is more conservative. And that's why you can see the inflation from 385 to 474 is more. But O'Brien Fleming is more realistic. It gives, well, more conservative boundaries at the start, but less conservative or less stringent boundaries at the end of the study. And you can see from 385 it goes to 393. But however, whenever you have this group sequential notion in mind or in the study squeezed in, you will only inflate the number of patients that is required. This is also an example taken from this book by Shaw and Shaw. I have referred to this book in the reference section as well, and the calculation can be found in that book too. I'm not going into the details of these calculations. So here I will end this presentation. Most of these examples are in this book, and one example is taken from a neuroscience study by Novartis, and You can go to that study by clicking on the link on the corresponding slide. The calculations that are done and shown in this presentation are done in R. So there are three types of calculations, one for continuous endpoint. I have the corresponding R codes for continuous, for proportion, and for the time to event. And thank you for your attention. I will stop here and get some questions.
Very good. So thank you so much, Subayan. And really interesting to see the simulation based approach, time to event, and so on and so forth. Now I don't see much questions in the chat. There is, I think, one question is the difference between group sequential design and adaptive design.
Well, adaptive design gives you, I mean, there are adaptive designs where you can keep the window open for what to do, that is open to you, but in which scenarios you will do, that you can mention in the protocol on the SAP. But in general, for the group sequential designs, you essentially state your boundaries or the decisions that you will take. For adaptive designs, let's say after 1 year, you have assumed some treatment difference for your study, but you see after 1 year that your treatment is not that good. So now you don't need 500 patients, you might need 700 patients. But that is not coming from the boundary that you set up for the group sequential design. That is coming from, there is a notion called reassessment of sample size calculation. There are blinding and non-blinding counterparts of it. But these two goes hand in hand when you essentially trying to change something in between the trial. If that answers the question.
Okay, very good.
Are there anything else? No, I don't see anything. After 12:14.
Okay. No. Well on time. Thank you so much. Over to you, Preeti.
So I'll not take much time. Kaushik, over to you. So Kaushik can do the vote of thanks.
Thanks, Preeti. Good afternoon, everyone. So although we are nearing the lunch time, but I'm very excited to feel the energy and the positivity around, especially the questions of the participants from the Q&A session. I sincerely request for a few more minutes of your attention. A webinar would not be concluded without an expression of appreciativeness. On behalf of Iscr Biostatistics Council, I would like to extend a warm vote of thanks to all of you for joining us today for this enlightening second webinar of Biostat. We are truly grateful to you for your presence and active participation. First of all, we would like to express our sincere gratitude to our extended speakers, Dr. Mahendra Vijarnia, Neha Agarwal, Arthri Kanojia, and Shubhayan Mondal, for sharing their knowledge and expertise on today's topics with all of us. Thank you for your valuable insights and engaging presentations, followed by the respective Q&A session, through which the participants got valuable insights about the importance of clinical trial design and features, rationale of randomization and masking, and sample size estimation. We all would agree that to conduct a webinar seamlessly will require proper planning and execution. So finally, I would like to thank our sponsor Iscr and Iscr Biostatistics Council Organizing Committee for their generous support. It has been indeed their staunch commitment that enabled us to organize this webinar and deliver such valuable content to our participants. I sincerely appreciate our technical team for their diligent work behind the scenes in ensuring a smooth and glitch-free experience in today's webinar. Last but not the least, I convey my gratitude to the audience for their time and attention. Your presence, interest, active participation through thought-provoking questions have made the webinar a decisive success. We sincerely hope that the knowledge and insights gained today will be valuable to your career journey and looking forward to future engagements. And while mentioning about future engagements, which were displayed during the break time, I'm glad to announce that we would have subsequent third and last Biostat webinar under the same umbrella on 8th of November. In addition to that, we look forward to hosting you in the third and last webinar of application of R organized by Statistical Programming Council on 18th of October. Thank you all once again and goodbye.
Thank you. Bye-bye. Have a good rest of the day. Happy Friday and have a lovely weekend. Bye-bye.
Bye.
Bye.
Bye.